Okay, well, welcome and thank you all so much for coming. I've been really excited about this talk. It's one of my favorite things to discuss, actually. So I'm delighted to have a captive audience here. Many of you I know and some of you I don't. So just a little bit about me, briefly. My name is Cotter. I'm very honored to serve as the Executive Director for Sierra Psychedelic Society and their sister advocacy organization, Nevada Coalition for Psychedelic Medicines, which is a C4, 501c4, and has been instrumental in helping to pass the legislation in Nevada, SB 242, with the help of our grassroots community, which has been incredible, and many advocates, some of whom are in the room today. I would venture a guess that most of us, if not all of us in the room, are here because we care deeply about healing, and we're very invested in healing for those that we love and for our community. And I find that with psychedelics, we have an enormous opportunity. I'm personally called to this work because psychedelic medicine has been the single most profound tool for me in terms of my own healing and personal growth. That said, it's important, before we dive into all of the amazing things about psychedelics, they aren't safe for everyone to be using, and particularly with the classic psychedelics, we need to be careful with a predisposition for schizophrenia, or if there's a close family member that has schizophrenia. Also, if there's a propensity for psychosis or bipolar disorder. So there are some abundance of caution. There's not a lot of research on this because the clinical trials will disqualify people that have those indications. But there is more work being done on this, and there is also anecdotal data to support that they can be helpful in certain circumstances. But generally saying we don't take any chances for that. So before we dive into the neuroscience, I just wanted to say that, as I was mentioning earlier, we proudly have now put Nevada on the map along with 21 other states that have active legislation. There are up to 16 or 18 cities that have decriminalized entheogenic plants and fungi. Colorado and Oregon have statutes for regulated access to psychedelic treatment, and Australia has now legalized MDMA and psilocybin for therapeutic use. So there is a global community of advocates and policymakers and researchers and scientists who are working very hard to make it possible for people to access these treatments. The history of psychedelics is a long and winding road, and we don't have time to go into that tonight, but suffice to say that there was 3 decades of research prior to the Controlled Substance Act of 1970. Very promising research. 40,000 research participants, 1,000 papers published, 6 global conferences, and then with the Controlled Substance Act, all of the research was shut down. And unfortunately, the psychedelic compounds, for some understandable, also very unfortunate reasons, were placed on the Schedule I list alongside some incredibly harmful substances like heroin and crack and whatnot. So in the meantime, we've been battling 50 years of conditioning that these substances, these compounds, are very dangerous, and that includes, a boatload of propaganda and some misinformation, and also some, I'm sure, well-intentions along the way, too. In the late 1990s, Rick Strassman, with his work with DMT, and then early 2000s, Roland Griffiths pioneered the way for what we call the third wave for the psychedelic renaissance. Roland Griffiths with his work at Johns Hopkins with psilocybin, and we now have over 20 years of research, a boatload of clinical trials that are astonishing, showing that these compounds are highly effective for treating anxiety, depression, PTSD, addiction, OCD, eating disorders. There are clinical trials at Johns Hopkins now for Alzheimer's and Parkinson's. And I know that a lot of us in the room would love to dive into the specifics of all the clinical trials. Since we don't have enough time to do that, I will be referencing the research as we go, but feel free to look on our website because you can check out all these trials to see the results, how amazing they are. So before we get into the neuroscience, because I'm going to take us on as much of a deep dive as we can in less than an hour and a half. I'm going to cover just some basic overviews about the brain and some review about cell biology. It's been a while for most of us. Oh, and before we do that, I was talking about the dangers and the harms of these substances. We have Dr. David Nutt to thank. He's the, I'm getting a little feedback, but one of the leading, if not the leading, neuropsychopharmacologists in the world, and he spent his career studying every kind of drug that you might have and the effects on the brain and body. And so he developed this harm scale, which is based on the metrics of harm to self and harm to society. And as you can see, psilocybin mushrooms are on the far right, the safest that you can be. Not far behind is LSD, MDMA. Ketamine is in the middle. Ketamine does have an abuse potential, and when it's chronically used, usually in recreational settings, it can lead to severe bladder and kidney issues. But in clinical settings, it is incredible for treating acute depression. So, as you can see, alcohol is 12 times more dangerous than psilocybin mushrooms. So moving on to the brain. The brain, as we know, is a highly, highly complex system. It's estimated that there are 100 billion neurons, and each of those neurons has up to 10,000 different connections. And the way that our brain is structured is we have this oldest, deepest part of our brain, which is the reptilian brain, which is in this subcortical area. Then we have the mammalian limbic part of our brain that is responsible for our emotions and our stress reflexes. And then we have the neocortex, which is the most evolved part of our brain that is responsible for abstract thinking and conceptual thought and rational thinking. And it sets us aside from other primates and all the other mammals too. And so we'll be talking about the neocortex, so I wanted to bring an image of this up. So when if we zoom in and we have a neuron, so a brain cell, essentially, on the level of a spatial scale, we'll have a neuron and then we have neurons that are working together, which form neuronal populations. And then when these populations are working in conjunction, they form brain regions. And then these brain regions that are working together form these higher order networks in the brain, like the default mode network, which we'll be talking about too. So here's an image of a neuron, probably familiar, but we have the cell body, we have the tail, which is called the axon, and then we have these branches, the dendrites, which are critical for communicating between different neurons. And then on the branches, on the dendrites, we have little spines. And it's not important to remember the names of the spines, but I did circle the one that's called the mushroom spine, which is because of the shape of the structure. It doesn't have anything to do with psychedelics, but I was tickled to see that. And so when we're talking about neuroplasticity and structural and functional plasticity, it's important to keep this image of the branches and the dendrites and the spines. The branches are the dendrites. So neurons come together to communicate, and there are some exceptions to this where neurons will actually physically touch, but generally speaking, neurons don't actually physically touch. They come together at a synapse. the space between them, also called the synaptic cleft. And they communicate by sending chemical signals, neurotransmitters, neuromodulators, across the space, the synapse, and then they will bind to the receptor. Here's a very rudimentary understanding, but a ligand is like a neurotransmitter. That's what fits into, it's like a key in a lock, like a cell membrane. Here's a slightly more sophisticated drawing of the ligand, which will fit into the receptor, and then we have very complex intracellular signaling processes that lead to response, generally making the cell more excited or less excited. And then this gives us a little bit more of an image where the neurotransmitters are generally packaged in these bubbles called vesicles. And when the neuron receives an impulse, it will move to the membrane, will fuse with the membrane, and then it will spit out the neurotransmitters or the neuromodulators. They'll move across the synapse. And if they happen to fit in their appropriate receptor, they'll bind to that receptor. So all this is critical to the discussion about the classic serotonergic psychedelics. And when we say serotonergic, it means it's acting on the serotonin system. Contrary to popular belief, the classic psychedelics don't elevate serotonin. Their chemical structure, and as you can see, just to back up, these fall in a class called tryptamines, where generally includes psilocybin, DMT, LSD. Their molecular structure is very, very similar to serotonin, as you can see on the upper left, and then the lower right hand, psilocin, or psilocin, however you want to call it, is a metabolite of psilocybin, so psilocybin breaks down into psilocin. So they're able to essentially mimic serotonin and bind to various serotonin receptors. Just put it down there. And what happens, so serotonin has a huge variety of things that it does in the body, which if there's time and interest for the questions and answers later, we can circle back to that. But they do bind to multiple receptors, but they have a very high affinity for one receptor in particular, which is the 5-HT2A. 5-HT just refers to serotonin, 5-hydroxytryptamine. And when it binds to this receptor, it induces a high degree of neuroplasticity. There's a lot to say about neuroplasticity, and I know there's a lot on this slide, but this is recent research that came out in February where it's not just that they bind to the 2A receptor, but there are 2A receptors inside the cells, intracellular 2A receptors, that are endogenous serotonin, meaning the serotonin that we naturally make. Oh, thank you. The endogenous serotonin that we naturally make doesn't permeate the membrane. It normally just binds to the receptors on the outside of the membrane. But the classic psychedelics have the ability to cross the membrane. I think it has to do with lipids, and they're a little greasier and they're able to permeate the membrane. But their action inside with these intracellular 2A receptors is huge for the neuroplasticity that comes from that. And just a note about neuroplasticity. Neuroplasticity is neither good or bad. With all the hype in the media about psychedelics and neuroplasticity, it's really easy to assume that what we're going for is neuroplasticity. More is not always better. We don't actually want our brains to be highly neuroplastic all the time. What we're going for is adaptive neuroplasticity. And with this 2A receptor, there was a paper that Robin Carhart-Harris, who is one of the leading psychedelic researchers in the world, he published this in 2020. It's called Pivotal Mental States. And he's talking about this particular 2A receptor, all drugs aside, how it acts in our body. It's a evolutionary biological mechanism that when we're under acute physiological or psychological stress, there's a very high release of serotonin, and it binds to this receptor, and it upregulates the neuroplasticity hugely. And this is meant to help us adapt very quickly and learn very quickly so that we can cope with these extreme circumstances in our environment and survive. So it's a different kind of neuroplasticity than, the everyday trying to learn how to play the piano, muscle memory sort of thing. It sets up a really high-speed circuit. It's implicated in PTSD, as you can imagine, and so we'll talk about that later too with MDMA. But what we have is an opportunity where we're able to activate this receptor when we're not in a life-threatening situation, hopefully. Well, safety, due caution, and setting have been cared for in terms of psychedelic experience. And we get this upregulation of neuroplasticity in a way that allows us to adapt. And there are several things that are going on with this. And it's important to the conversation, kind of jumping straight to this, but the way that it works, and I'll tell you what this is about and then I'll explain. What we know about chronic stress-related mood disorders like depression is that there tends to be an atrophy of the dendrites and spines on the neurons. And so you can see here is a neuron on the left. It's actually called a layer 5 pyramidal neuron, which is the actual primary neuron that these 2A receptors are on. Not important to remember. However, you can see under normal circumstances it has all of its branches intact, and then after stress, they start to diminish. And then this is zoomed in where you can see the spines on the dendrite with stress, much less of these spines. And so when we have a plastic psychedelic, and I'll get into how this plasticity occurs, but there have been several studies, and this one is out of UC Davis with David Olson's lab, where they applied LSD to the brain tissue of a rat. And the VEH refers to the control. DOI is a research psychedelic that agonizes the 2A receptor, so it's not one that people are usually taking. DMT, you can see there's an increase on the spines on the dendrite, and then a huge increase with LSD, which is incredibly promising. So what's going on here, here's another little drawing to give you an idea of healthy brain, branches, depression, atrophy, psychoplastogen, which is a psychedelic that induces plasticity, allows us to regrow some of these branches. This is also huge with ketamine, which we'll talk about later too. So with the 2A activation, what happens is, I don't know when I said thirsty. It's the excitement of the talk. So we have a psychedelic that mimics serotonin. It binds to this receptor and it modulates various neurotransmitters in the receptor, particularly one called glutamate, which is the brain's primary excitatory neurotransmitter. So the brain operates with a system of accelerators and brakes, which are necessary. Some neurons make other neurons more likely to fire. Some make them, the inhibitory neurons make them less likely to fire. And so when you have a net increase of glutamate, what it does is it is released into the synapse, it affects the receiving neuron, and it excites it, and then it triggers a number of intracellular processes, including an upregulation of BDNF, brain-derived neurotrophic factor, which is one of the primary growth factors in the brain. And BDNF does a whole bunch of things where it will, in the presence of glutamate, it will cycle a bunch of glutamate receptors to the surface of the cell. These are called receptors. It also has the ability to change the structure of the cell so it can grow new dendrites and new branches. And this is huge because when there are more dendrites and more branches, it increases the synaptic strength with other cells, which would stand to reason because they're able to communicate and there's more spines and dendrites. It also has the effect of this glutamate. Interestingly enough, normally when the glutamate is in the synaptic space, it'll affect the neuron and then it will be cleared and sucked back up into the neuron that released it. With the 2A activation, what it does is it spills out of the synaptic space in a very diffuse way, and it starts to affect all these other neurons in the vicinity, and it excites them as well. And so what we get is this sort of out of sync phasing where normally neurons might be firing together, and then they become excited with glutamate, more dendrites, more synapses, all this activity is going on, and it becomes a lot more random. And this is related to the term entropy that we talk about a lot with psychedelics, where entropy in this context means uncertainty or disorder. It's a term that comes from statistical physics, thermodynamics. It indexes the predictability of randomness over time. So if a neuron would normally be just firing steady as they go, and then gets excited, and then all the other neurons get excited, and they start firing out of sync, we get all this entropy, which is great. The reason that this is particularly important is because of the location of these 2A receptors. So I was hesitant to show this slide, but I'm fascinated with what they look like. So this is the neuron, these layer 5 pyramidal neurons, and as you can see, so layer 5 is relating to, we're depending on where you are in the cortex, there are different layers. So 5 or 6 layers. They have these really long tails, they branch out at the bottom, and then they have these pyramidal shapes that are interconnecting with all these other neurons. And these are found in a particular part of the brain called the association cortex. These 2A receptors are highly, highly expressed in the cortex, the neocortex, and the visual cortex, which isn't surprising because we get with psychedelics visuals, closed and open-eye visuals. And in this association cortex, it overlaps hugely with our default mode network. So what happens when our brain, in this hierarchical system with our brain, where we receive sensory inputs, this is referred to as bottom-up, and they have a feed-forward pathway, and our brain has to figure out how to translate this information into a more sophisticated concept, like this light, color, and shadow that I'm seeing is a tree, for instance. So the sensory input comes up, feeds forward from the bottom, and then we have these higher order, very evolved networks that are at the top of the hierarchy. It's called top-down control. They have feed-backward pathways, and they meet in the middle at the association cortex, which is responsible for integrating this information and sending it throughout the brain. So the association cortex, being so strongly overlapped with our default mode network, is huge to the function of the classic psychedelics because the default mode network is the area that is responsible for mind wandering, daydreaming, thinking to the past, projecting to the future, metacognition, which is thinking about thinking, the ability to reason about what other people are thinking, and rumination. And it often is overactive in conditions like depression and anxiety and addiction. And so when we get all this entropy in the default mode network, what it does is it disrupts the normal functioning of it. And networks, they refer to this as like being modular. So they're very sort of intrinsically connected within themselves, and they talk within themselves, and then there's a certain amount of talking to the other networks, which is more called like a global communication. And the default mode network is highly interconnected with all the networks as like a hub. So when it disrupts the normal activity of the default mode network, it essentially downregulates the activity within itself. But it takes the constraints off the communication with all the other networks, and it increases what's called global functional connectivity, where networks in the brain that don't normally talk to each other start talking to each other. So we get synesthesia, or we're hearing colors and seeing sounds. But it also may allow us to have access to memories that we normally repress. And one of the beautiful ways to think about this, there's a model. This is a little review about the association cortex. There's a model called the Rebus model, which stands for Relaxed Beliefs Under Psychedelics. Again, Robert Carter Harris published a paper, 2018, called the Rebus model, which is building on a 2014 paper called the entropic brain hypothesis, which we've alluded to with all this entropy. So the Rebus model is one where there's this leading perspective that with this hierarchical functioning in our brain, that this term hierarchical predictive coding, and what this means is that in order to function in the world when we have all this sensory input, all this stuff going on that would normally be chaotic and overwhelming, these more evolved networks in our brain find a way to bring some stability and predictability. It essentially, we form these, what they call priors, so they're prior beliefs, where, and this is very important, it's critical to our functioning. If we, based on our prior beliefs and our experience in the world, we know that, generally speaking, laws of physics, we drop a ball, it's going to go down instead of up. But this also happens with our experiences in life that can encode prior beliefs about ourselves and the world. And these can look like, I'm not worthy of love, people are not to be trusted, the world is not a safe place. And so these can become very deeply ingrained, and they're encoded in those pyramidal neurons and the default mode network. And so when we get this high amount of entropy and neuroplasticity, the Rebus model suggests that it's essentially relaxing these deeply reinforced neural grooves that have been existing, and it gives us a window to repattern. It gives us access to a much broader variety of brain states that we might not have access to if we're stuck in a space. And this critical window of plasticity can last up to weeks or longer. And so this is why it can be so important when we're talking about psychedelic sessions, the mindset going in, setting the intention, not just during the journey, because the peak of the plasticity for the psilocybin, for instance, falls about 6 hours into the journey. And then it will last for, nobody knows for sure, but the research suggests it's up to 2 weeks, potentially 30 days for some of these classic psychedelics. So you can see here, this is just a model about the top-down beliefs influencing the other part of the brain. The PFC refers to the prefrontal cortex. PCC is another part of the default mode network. Posterior cingulate cortex, not important to remember. But there's this reciprocal interaction where they're communicating with each other. And here's a more colorful image that is probably familiar to most, if not all of you, also from Robin Carter Harris's lab. This is based on the fMRI imaging with psilocybin, and it's representative of all of these networks in the brain that are communicating that don't normally communicate. So That was a lot of information. We're going to keep moving here, but I did mention in the talk description that I was going to delineate between 5-MeO and N N-Dimethyltryptamine. And I find this to be fascinating. So 5-MeO DMT, which is, it comes from the Sonoran desert toad, from a gland, and it has a very different subjective experience from when people normally refer to DMT. They're referring to N N-Dimethyltryptamine, which tends to have a lot of geometric visuals. We won't go too much into the difference of the subjective effects, but 5-MeO is also tryptamine. However, it has a very high affinity for the 1A receptor, serotonin 1A receptor. And this is, it will hit the 2A receptor at higher doses, but the difference between the 1A and the 2A with serotonin is they have sort of opposing mechanisms. The 1A is where our endogenous serotonin generally binds to, and it chills us out. Everyday stressors, not the acute stressors. This is also the receptor that conventional pharmaceuticals act on, and that is very interesting, and time allows, we can go into that too. But as opposed to the other classic psychedelics act on the 2A receptor, and where these receptors are located, so the 1A is very much associated with our limbic system and our stress response. And this theory comes from A Tale of Two Receptors. It's a David Nutt, Robin Carter-Harris paper, where they're outlining the difference between these two, the 1A and 2A. And so with 5-MeO acting on the limbic and the stress system, it's called passive coping, as opposed to the 2A, which is more in the cortex, and it deals more with emotional regulation. It's excitatory instead of inhibitory, and so this neuroplasticity falls more in these higher level networks. But it's, presumed that this 1A activation with 5-MeO actually has really powerful implications for our endocrine system and our stress responses. So I realize that was a lot, just in review. Little takeaways. Classic psychedelics mimic serotonin and modulate the 2A receptor. They increase functional and structural neuroplasticity. They do this by way of growing new dendrites and new spines. They strengthen synaptic connections. These are most highly expressed in the cortex, these 2A receptors. They also are expressed in some subcortical regions like the thalamus and the claustrum, and there's some theories around that. We can come back to that too if there's time. I want to talk about all the things, but I'm trying to be mindful that we probably all don't want to be until midnight. So they disrupt these higher order networks, like the default mode network, which allows us to have this interconnectivity throughout the brain, this global functional connectivity. And there's a malleability with these rigidly held encoded beliefs that we have an opportunity to basically repattern them. So, sorry, that was a slide that didn't get made. Moving on to MDMA. 3,4-methylenedioxymethamphetamine. MDMA falls in an entirely different class. It's a phenethylamine. It's also known as an entactogen, which means getting in touch within, or an empathogen, which induces empathy for self and others. And what it does is it doesn't mimic serotonin. It triggers a high release of serotonin and dopamine. It also releases, due to the methamphetamine component, norepinephrine, which contributes to focus. This is a very unusual situation in the brain because normally dopamine and serotonin are inversely correlated. But when you have this high release of serotonin, which is huge for relaxing us and bringing a sense of contentment here and now, massive reduction of activity in the amygdala, which is often considered the fear center. It does a lot more than that. And then having the dopamine which is not as much as the serotonin. The serotonin release is 3 to 8 times higher than the dopamine, but the dopamine piece is non-trivial and actually huge to this process, particularly when we're talking about MDMA for therapeutic use. The dopamine is also a feel-good chemical, so these two combined generally contribute to a profoundly good mood. That's not to say when you're working with trauma in a therapeutic session that it's all rainbows and bliss, because it's not necessarily, but the dopamine contributes because it's associated with reward and motivation. There's a sense of reward and motivation attached to engaging with the therapy. And when you're working with trauma, so I wanted to give this visual because MDMA also, there's a certain Navy SEAL in the room who's quite fond of this one. I find it very endearing. MDMA doesn't just release the serotonin and the dopamine. And it's fascinating how it does it. It's a two-pronged process that sort of blocks the transporter at the cell membrane, leaves more serotonin or dopamine or norepinephrine. It's a similar process for all of them. And then it interferes with the packaging inside the cell into the vesicle. So you have a lot of serotonin, for instance, intracellular space. When there's an action potential, all goes out. I have some slides if you want to go deeper on that. But it also binds to some of the serotonin receptors. The 1A receptor, which is associated, like we were just talking with chilling us out, kind of bringing everything down. And then here we got, I'll bring us through here, the 2B. And then this is what the image with the mice is about, is that the 2B receptor is associated with prosocial bonding. So we have this high degree of a desire to connect with others. And with animals, this is called adjacent line behavior, which means cuddling. And so this can be very helpful in a therapeutic setting where you're dealing with something that is your own personal crisis, and having the security and safety and the trust in the therapist. That is promoted in large by the 2B binding. And then we also have, it does bind to the 2A, much, much lower affinity, but it does upregulate some neuroplasticity, which can account for, a little bit of psychedelic colors pop a little more, but the walls aren't melting. But it's in a pathway, in the reward pathway. It's an area called the nucleus accumbens, and so there are these downstream effects of dopamine that contribute to the reward and the motivation. I know I sort of skipped past this, but it also releases a high amount of oxytocin and vasopressin. And you would assume that oxytocin, being a para-bonding hormone, would be contributing hugely to this, but the research is really mixed on this. Some of the studies that they found that it may not actually be so much the oxytocin. So with MDMA in a therapeutic context, where this gets really fascinating is that we now know that our memories are not stable, so they can be reactivated, we bring them up, and there's a process where they get reconsolidated back through the hippocampus. With trauma, like we were talking about before, there's this highly neuroplastic state, again, evolutionary system to keep us alive, and something super overwhelming to our nervous system or life-threatening happens. It is a very quick lay down the circuit. Even a glimmer of this thing ever happens again, you're going to be in high alert. And so it's maladaptive in this sense. but it also makes it difficult to treat PTSD because it's embedded in your nervous system. And there are a number of therapies, and I'm not an expert on trauma, but we do have some people in the room that are, you can always ask. But generally speaking, the talk therapies, like cognitive behavioral processing and cognitive behavioral therapy, are ones that are relying on essentially exposure therapy, whereby recalling the event in great detail over and over and over again, there's an attempt to decondition and uncouple the emotional response from the memory, sort of like Pavlov's dog, where, you know, initially the conditioned response, you get the food, ring the bell enough times, salivate, even when the bell is rung. We can leverage and harness this same circuit to decondition, but it can be a long and arduous task and really difficult if every time you're trying to reactivate this memory, there can be this overwhelming emotional response, so it can be reconsolidated the same way. With MDMA, what we have is an opportunity where our biology is working for us, so that when we bring the memory up, we have all of the feel-good chemicals happening, this massive reduction in anxiety, and when it's reconsolidated, it can be reconsolidated in a different way. And the research shows that it's not just enough to have the fear extinction on that circuit, the deconditioning, but you also have to attach a positive reframe, which inhibits the initial circuit. And that can be really tricky when you're dealing with a situation that was perhaps the worst experience of your life, to find a positive way to consider it. But what this can look like for people is focusing on their resilience, or look how amazing it is that I survived that situation, how I might be able to help other people, any number of things. And so essentially what we're doing is we're reattaching a different perspective to memory as it gets reconsolidated. So it's just a little message on that reconsolidation there. Also, this is why just taking MDMA by itself, which might be an incredible experience depending on the context, doesn't necessarily resolve PTSD, because it's what you're doing with that experience and how you're working with the plasticity and how you're working with the memory recall, how it can be reconsolidated through the hippocampus. So, this is a quote that I think is a really good example. This is from one of the participants in the MAP study. He says, one thing the MDMA facilitates is thinking about traumatic experiences in a neutral, safe manner. I could objectively think about them and talk about them. Then it seems those memories are put back in their place in the brain in a different configuration. A configuration that does not cause as many problems, such as bad dreams, intrusive thoughts all the time, or having horrible insomnia. This has continued to this day, 1.5 years after the MDMA session. And breaking news, 2 days ago, yesterday actually, published in Nature Medicine, which is one of the top scientific publications, the MAPS phase 3 trials, the second of their randomized placebo-controlled trials, showed that there was significantly improved PTSD symptoms compared to the placebo over 18 weeks. And as you can see there, 86% of the participants treated with MDMA-assisted therapy achieved a clinically meaningful benefit, and 71% of them no longer met the criteria for PTSD by the study's end. So this is phenomenal. And yes, 40 years of research, Doblin pioneering this, working so hard, and as you know, FDA had granted breakthrough therapy designation and making huge groundwork for this. So Thank you for celebrating. I did want to say, if in case somebody isn't suited to MDMA, where it's not the kind of thing where they can, it's safe for them to take that as a psychedelic, all hope is not lost, because there are a lot of resources for treating trauma, and so I've listed some of them here, and be happy to share after, but there are some incredible people doing work in this field, and there are other ways to treat trauma too. This is another slide that I thought would look more pleasing to the eye and forgot to take the other one out. Moving on to ketamine. So, ketamine is a dissociative anesthetic that has psychedelic properties. It is acutely helpful for treating, profoundly helpful for treating acute depression and suicidality. It's more short acting. It tends to last 3 days to about a week. And ketamine is one of the most fascinating of these substances, and I will say that I have gained an entirely new respect for this compound upon learning about the pharmacology. There are so many different layers going on with ketamine that it is incredibly fascinating. Ketamine, as I mentioned earlier, promotes a huge amount of neuroplasticity. And also, I think I mentioned this, but it's been shown with studies that even after one ketamine session, you can regrow the dendrites and spines that have atrophied. The plasticity is associated with these antidepressant effects. And so this is where it gets super interesting, because ketamine is what's called an NMDA antagonist, and NMDA, N-N-Dimethyltryptamine, is a glutamate receptor. And so we were talking about glutamate and the excitatory processes and that contribute to neuroplasticity. These NMDA receptors are actually critical for plasticity. And so it was really puzzling to the researchers why, if it's having these antidepressant effects, as well as plasticity, but it's blocking the glutamate receptor, it didn't make a lot of sense. And some researchers, thankfully, kept looking into this. And what they discovered is that the NMDA receptors that it blocks are on inhibitory neurons. So what happens is it's inhibiting the inhibition. So it's like a double negative, where it results in a high degree of excitement, and you get this glutamate surge, and the glutamate spills out, like we were talking about. But normally when there's a whole bunch of glutamate, there's this internal mechanism of the cell that is there to pump the brakes. If there's too much excitotoxicity and everything's firing too much, and it spills out, and it hits the NMDA receptor around the side, it enters, but it enters in a very non-directed way and essentially pumps the brakes on the plasticity and can promote cell death in order to rein everything in. But because ketamine is blocking the receptors around the side as well, around the side of the cell, you get this double amount of neuroplasticity. So I thought it might be helpful for a visual on this. So under normal circumstances, we have this inhibitory neuron over here, and it's sending neurotransmitters across the space, and there's a little bit of glutamate release, but it's basically business as usual, and there's not a whole lot. And then with ketamine, it blocks this inhibition and takes the reins off, and then we get this massive amount of glutamate that cycles down. It then goes through that process where it upregulates BDNF. BDNF grows new branches and spines, cycles new AMPA receptors, which are also glutamate receptors, to the surface. BDNF can also, ketamine can actually mimic BDNF. and bind to BDNF receptors. And additionally, there's rodent studies where there's this part in the brain called the lateral habenula, which is known as the disappointment center, where if you're expecting a reward and you don't get it, and you're disappointed, this is where it gets activated. And what ketamine tends to do is it regulates the activity and it reduces this activity in the disappointment center. And that's relevant to depression because the lateral habenula can be really overactive with depression. And then the plot thickens because it turns out that ketamine also acts on the opioid system, which for a hot minute people were wondering, maybe this is having a lot to do with the antidepressant effects. The opioid system, their receptors, they name them after Greek letters. So ketamine acts on several of them, the mu, kappa, delta, particularly high affinity for the mu receptor. And so there was a study that came out not too long ago that made quite a splash where they used an opiate antagonist, naltrexone, to block the mu receptor. And it didn't change the subjective effects in the moment of ketamine. Ketamine still had really, it had, ketamine effects. But it did seem to inhibit the antidepressant effects after the fact. However, there have been several other studies that have tried to replicate this, and they weren't able to do that. So there's a lot of more research is needed, and so there's a big question mark as to how this is participating with the entire equation of ketamine. But I think that it's fair to say probably a huge combination of all of these factors that are going into the treatment for ketamine. Okay. Surprise. I should probably have the presenter interview going on so I know what's next, but I'm trusting in my earlier self. So, what I did want to say, after ketamine in a nutshell, is that we talk a lot about all these theoretical concepts like global functional connectivity and structural neuroplasticity and entropy and so on and so forth, but how does that really translate to our healing journeys? And for me, I have found that psychedelics have been an invitation, essentially, to really get to know myself and to embark on a journey. And sometimes the journey is punctuated by these blissful, expanded moments of deep interconnectedness and merging with frequency of can best be defined by love. And sometimes these experiences require me to dig really, really deep and draw on my own resilience and strength in the face of adversity. So I always go back to the metaphor of the hero's journey, which for those of you that attend the integration circles, this is familiar territory, but I take great solace and courage and inspiration in knowing that we're on this journey where we have these peaks and valleys, these mountaintop experiences, and these dark nights of the soul. But there's never one of these that defines the entire plot arc. And so when we have these moments where, in a psychedelic journey or life in general, because it's kind of all the same thing, depending on where you're zoomed into, and we have to face either literally or figuratively the monster or inner demon. And I couldn't resist bringing in some Tolkien for Lord of the Rings fans, so there was a spoiler a moment ago, but here we have. This is profoundly archetypal for me, where we have Gandalf with the Balrog, and ultimately this is defined by a rebirth, a death and a rebirth. And the other scene that is always so present to me is this one, Frodo in Shelob's lair, where we're reminded of our inner light, that no matter how difficult something is, we have caught these glimpses of our own divinity, and we carry that with us. The other metaphor that I really like is the one about is video games. So this is hilarious because I'm not a gamer at all, but I love to think about our life as, you know, we level up to the next level, and maybe formerly it was smooth sailing because we figure out all the things and knew what points to grab and this and that, get into a flow state. We graduate, end up in a completely unfamiliar landscape, incredibly challenging, and get kind of knocked down over and over again. But I'm reminded that these are very rich, important experiences where we're talking about neuroplasticity, and we're talking about learning, and we're talking about finding a way to level up. And when I was pondering how funny it was, because I haven't actually played a video game since 1986, when Super Mario Brothers came out, and my dad bought us Nintendo for Christmas, so I was pulling up an image, and was delighted to see these amanita mushrooms. And you know, talk about early childhood conditioning. They've always since beaten into right into the punch. So, you know, in closing, before we open it up for questions, I would just like to say, you know, essentially, I find that for myself, it's with deep reverence and wonder that I endeavor to comprehend as much as we can a rudimentary understanding of the brain and how it mediates these altered states and this deep healing. And furthermore, how it allows us to shed our stories about who we are and who we're not, and what the world is and what the world is not. And at the end of the day, I find that it allows us to be the architect of our own narrative through these beautiful plastic processes. So thank you all so much for listening, and we can take some questions, and you've been very attentive, and really appreciate you being here tonight. Is there anything that, I'm not a neuroscientist, so it's possible that you'll ask a question that I don't have the answer to, but I'll find out, because I know some neuroscientists that I can ask. Art.
I wanted to ask you about the serotonin depletion that follows MDMA use in particular, and if there's a valid concern that it might undo or redo the positive memory reconsolidation that happens during the session.
I think that is an excellent question. So Bart is asking about the serotonin depletion that happens following an MDMA ceremony. Thank you so much, Taylor. And whether that could potentially affect the memory reconsolidation. My best guess is that it probably would not be a primary concern. I think it would be, I mean, you have to be careful with MDMA, because when we do have the high release of serotonin and dopamine, there's a, you know, there's a dip after, and then our brain recalibrates. So it's something where you want to make sure there's enough time between sessions to do this. But I don't think that the dip in the serotonin afterward would necessarily be directly linked to memory reconsolidation. But I will certainly ask on somebody about that. I think that it would probably, there would be a net positive effect. I've had an experience where I was struggling and was a little hesitant to do an MDMA session because if you're already struggling with some depression and anxiety, and I was worried about the dip after, but I found that the therapeutic work that I was doing during the session actually was so profoundly helpful that whatever neurochemical dip that followed wasn't what I was expecting. It was actually super helpful. It's a great question. Bob.
I'm interested in conditioning and conditioned fears, and you could call conditioning learning, a kind of learning that so many fears we wind up dealing with around anxiety, phobias, trauma, they all belong to the same family in a sense. And so we learn them as we grow up through our experiences, and perhaps through war and other experiences. And so many of the benefits that I've read about and heard about through the use of psychedelics seem to all have a kind of a common characteristic of lessening fear. And the one that we hear about over and over again is fear of death. Many people, especially near-death experiences, no longer apparently have any fear of death, which is kind of amazing, that fear. So anyway, I guess I've come up with a little hypothesis of my own that one of the general benefits of psychedelics, I'm not being specific now about which one of the general benefits comes from a kind of deconditioning and a kind of letting go of some of these things that we've learned at some level where we might still have the memory, but the emotional learning is way, way, way down and whatnot. And so I just want to throw that out for you. Psychedelic therapy as a deconditioning therapy. Does that make sense to you?
Yeah, absolutely. And I would, for those of you that may not have been able to hear, Bob was talking about psychedelics as a form of deconditioning, which is exactly, encapsulates so much of what we're talking about tonight. One thing that I would qualify about psychedelics reducing fear, I think large trajectory as we do this work, like, end-of-life anxiety, all of that, absolutely has been my experience. However, it's normal and very common during psychedelic journeys to have fear and anxiety in the moment. And so I think that's where it's important to know going into it that it's okay if these experiences are characterized by moments of fear in the moment, but then after the fact, doing that deep work and struggling through and facing the fears that come up is where I think the beautiful results can happen long term. Does that make sense? Oh my gosh, James.
It is James.
Hello.
So I have a lot of issues, a lot of us do, I'm sure, but there was a childhood stuff, there was other things, and then when I was in the military, stuff happened that sort of exacerbated everything. And I went through all of these therapies, CBT, CPT, blah, 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 acronym war. And what I found was that I was getting all of these programs as if I were a computer, but the hard drive was damaged by all of those experiences. So I started ketamine in December, and the experience of it is that I've got a new computer that can run these programs so much better, and I can access things that were not available, on my Commodore 64. But I'm curious, I'm only doing ketamine, and it does, it's not a magic pill and go. I love the fact that you brought up the idea that it allows you to access these programs that your computer could not run prior, because it was just not up to speed, literally. I'm curious if you can use them in concert. Like, I see them individually, like here's the information on ketamine, here's the information on MDMA. Has there been any work done where they use them together?
Yeah, absolutely. And I know that, you know, do you guys want to take this? I don't want to speak out of turn about therapy practices because I'm not in therapy. I know that I personally have used in concert, but we do have experts in the room that.
I don't know of anything above ground, any studies above ground that use them in concert. The underground has been using them in concert for decades with really good, with set and setting and everything is as perfect as you can do with great results. I'm sure there's some outliers that are traumatized because that's, you know, is what it is, but it seems to be.
I just saw it. I'll have to go back and look, but Johns Hopkins was just releasing that they were going to do a study combining the MDMA and the ketamine. So it's starting.
Well, that's so interesting. I didn't know about a study combining MDMA and ketamine. I do know that there's a lot to be said about combining psilocybin, for instance, and MDMA, where you get the high neuroplasticity from the psilocybin, or commonly hippie flipping, or with LSD, candy flipping. So this has been around recreationally for a while. I know that underground therapies will often combine them together because then you have the ability to, like, with the classic psychedelic, we're going very deep in it's a very different kind of subjective experience, and sometimes that can be anxiety producing. I think that like a little bit of an insurance policy, if MDMA is present because of how we talked about with all the feel-good chemicals, but then you also get some of those other receptors hit too. So I think that's an excellent question, and the reason, part of the reason I preferred is I have relatively limited experience with ketamine myself, and so I wouldn't consider myself at all equipped to weigh in on practices where it's combined, but I'm very curious to know. So we have a.
So,
oh yeah.
I'm not a clinician. I'm completely underground. I look at plant-based medicines. I know that ketamine is a blocker. Plant-based medicines can actually inhibit you getting full experience in the journey. As far as MDMA, when you combine them, just on a recreational thing, I have beautiful experiences. We take MDMA and we call it baby flipping, basically mushrooms. And had some really beautiful experiences combining those and just really good work with marriage life, actually some marriage counseling together, and just together with the intention of getting through some stuff together, and we did that. But I know that what I've experienced and what I've seen in the plant-based medicines, I watched fungi and stuff like that, but ketamine actually is an inhibitor for the experience to happen because ketamine is disassociative, and it's not necessarily a psychedelic with debate on this. It's not in the family of psychedelics, so I think it's not recommended to it's not going to hurt you, but I don't think you're going to get the experience if you're doing plant-based medicine. I'm not sure about the MDMA and ketamine. I've never experienced it myself. But as far as they go, it's good to kind of clear out ketamine before you get into plant-based medicine so you can have the full effects. That's my experience.
Thank you for weighing in on that. Thank you. Yes.
So some providers like to talk about TMS therapy as a way to deal with depression. I was curious, your thought on that, and of all the various options that you laid out, is one of those approaches comparable or similar effect to TMS, which is really expensive?
Yeah, I is it? Okay. Because I'm aware that Nolan Williams has done a lot of work out of Stanford with TMS, and to be quite honest, I haven't done any deep dives on that, but Taylor is suggesting that could be helpful in combination, which would make sense. Yeah, that's a great question.
I have clarification on this question. I have no idea what TMS therapy is.
Transcranial magnetic stimulation. And in fact, Taylor, feel free to elaborate if there's.
Essentially, you get like a helmet put on and you're hooked to this big machine and it sends little magnetic zaps into your brain to stimulate the production of serotonin.
Super fascinating. I know, oh, go ahead, yeah.
Have there been any large study or even anecdotal studies, I guess, on the effects of psilocybin with ADHD?
I haven't heard of any, but I wouldn't be surprised. I think I may have read that there are some in the works. That would be really interesting because, yeah, it would be, I wouldn't be surprised if there are some happening, and I'd be happy to look it up. Curious to know myself. Do you know?
Anecdotal, I find like microdosing LSD to be the most helpful. More so than psilocybin for treating with ADHD, but everyone has a unique experience.
So, in case you couldn't hear, Taylor was suggesting anecdotally that there seems to be a lot of help with microdosing LSD in regard to ADHD, more so than with the plant medicine. But you know, everyone's chemistry is different, so that might be something to consider.
We read the book A Really Good Day, which kind of discusses that. My wife is very interested in the topic.
Any other questions? You guys have been great. Thank you so much. I know that was a boatload of information, and we appreciate you tracking with me on that. So, thank you for coming tonight.
Thank you.